Dear learners, in this video I will discuss about vaccine, introduction, types and modern strategies. Introduction. The term vaccine was coined by Louis Pasteur to commemorate first successful immunization against smallpox by Edward Jenner. Etymology of vaccine comes from vacca, meaning cow. Vaccine is a suspension of whole, live or inactivated or fractionated bacteria or viruses that have been rendered non-pathogenic and is given to induce an immune response and prevent disease. Vaccination is the deliberate introduction of an antigen into a host with the intention of inducing immunity. Characteristics of ideal vaccine. First is provide long-lasting immunity. Second, should induce both humoral and cell-mediated immunity. Third, should not induce autoimmunity or hypersensitivity. Should be inexpensive to produce, easy to store and administer. Fifth is vaccines must also be perceived to be safe. Types of vaccines. First is natural live vaccines. Preparations include natural non-pathogenic organisms. Currently, these vaccines are rarely used. Problems include their ability to mutate and convert into pathogenic forms. Example, first is cowpox that has scientific name Vaccinia sp, the virus used for immunization against smallpox. Second, simian rotavirus is used in vaccinations against gastroenteritis. Second, vaccine killed or inactivated vaccines. When it is unsafe to use live microorganisms to prepare vaccines, they are called killed or inactivated. These are preparations of the normal infectious pathogenic microorganisms that have been rendered non-pathogenic, usually by treatment with using heat, formaldehyde or gamma rays so that they cannot replicate at all. Efficiency of vaccines vary greatly. Examples include Salk polio vaccine, rabies vaccine, pertussis vaccine. There are some advantages. First is no mutation or reversion to wild type forms, provides sufficient humoral immunity, heat stable, can be used in immunodeficient patients. Disadvantages include weak cellular responses, repeated booster doses are required, higher cost, disease occurrence when inadequate inactivation of the virulent organism. Third, live attenuated vaccines. These vaccines are composed of live attenuated microorganisms that cause a limited infection in their hosts, sufficient to induce an immune response, but insufficient to cause disease. Attenuation or weakening is done by growing pathogens in a foreign host, such as embryonated eggs or tissue culture cells, for a prolonged time. Examples include BCG, Bacillus calmette-guerin, Sabin polio vaccine, MMR, measles, mumps, rubella, varicella zoster vaccine, yellow fever vaccine. There are some advantages. First is induction of memory, cellular and humoral immune responses. Second, less amount of antigens must injected to induce protection. Third, repeated booster doses are not required. Fourth, oral preparations can be possible and these are inexpensive. Disadvantages include first, very rarely revert to its virulent form and cause disease. Second, live vaccines cannot be given safely to immunosuppressed people. Third, proper storage is critical. Fourth vaccine, toxoid vaccines. Toxoid vaccines are made up of toxoids, which are chemically modified toxins that have lost their toxicity yet are immunogenic. Examples: diphtheria vaccine, tetanus vaccine. Fifth vaccine, polysaccharide or conjugate vaccines. These vaccines are primarily developed against bacteria which contain capsular polysaccharides. Polysaccharide antigens are T independent, they generate short lived immunity. By covalently linking or conjugating the polysaccharides to protein carriers, they are converted into T-dependent antigens and protective immunity is induced. Examples include Haemophilus influenzae B vaccine, vaccine for pneumococcal pneumonia, and meningococcal meningitis. Sixth, recombinant vaccines. These vaccines are produced using recombinant DNA technology or genetic engineering. Recombinant vaccines are those in which genes for desired antigens of a microbe are inserted into a vector. Examples include hepatitis B virus surface antigen vaccine, BCG vaccine strain engineered to express genes of HIV-1, etc. There are some advantages. Vectors are not only safe but also easy to grow and store can be chosen. Antigens which do not elicit protective immunity can be eliminated from the vaccine. Disadvantages include cost of vaccine production is high, care must be taken to spare immunodeficient individuals. Seventh is DNA vaccines. These vaccines are still in experimental stage. DNA is injected into the muscle of the animal being vaccinated, usually with a gene gun that uses compressed gas to blow the DNA into the muscle cells. DNA can be introduced into tissues by bombarding the skin with DNA-coated gold particles. Examples include rabies vaccine, influenza vaccine, HIV vaccine are currently being explored. Here is the figure. Mode of action of DNA vaccine. DNA vaccine is injected into the muscle and within the host, DNA vaccine is actively functioned to induce strong immune response. There are some advantages. First is easy to manufacture in large amounts, stable and easy to transport, DNA sequence and hence antigen can easily be changed. Mixture of plasmids could be used to form a broad spectrum vaccine. Disadvantages include insertion of foreign DNA into host genome may cause cell to become cancerous. Second, danger of autoimmune responses. Third, cannot be formed against polysaccharide antigens. Modern vaccine strategies. There are several new vaccine strategies as follows. 1. Synthetic vaccines. These are preparations of known peptide sequences of protein antigens that can be chemically synthesized in a laboratory. Vaccines against AIDS, malaria, schistosomiasis, hepatitis B are currently being experimented. Problem is they are poorly immunogenic. Here is the figure. You can pause to see it. Second, microencapsulation delivery system. Antigens are placed inside a biodegradable microencapsule and introduced into the body of the recipient. Antigens are released from a capsule either when the capsule is phagocytosed by macrophages or dendritic cells or via slow leaching. Delivered orally or nasally, directed against influenza tetanus. Problem is also present in case cellular immunity is not induced. You can pause the video to see the figure. Liposomes. Liposomes are teeny spheres contain phospholipid bilayer. Interior of the liposome is aqueous in which soluble antigens are enclosed. When introduced into the recipient, liposomes are fused with the cells and released antigens into the cytosol of the recipient cells, directed against hepatitis A. Here is the figure. You can pause to see the video. Fourth is immune stimulating complexes or ISCOMs. ISCOMs are liposome-like structures consisting of outer coat of lipids mixed with detergent and a glycoside such as QIL-A. This can stimulate both humoral and cellular immunity, hence oral vaccines are widely used. directed against measles. Here is the figure. Fifth is solid matrix antibody antigen complexes, or SMA. Monoclonal antibodies are covalently linked to solid matrices. This then antigen binds to these complexes, form SMA. After introduction into the recipient, this can elicit strong immune response directed against several infectious diseases. Here is the figure. Thank you, viewers. Thank you for your support. If you like, please subscribe and share my channel. Thank you.